Choung from the Mayo Clinic in Minnesota, and the talk is entitled Interactions Between Gut Microbiota and the Immune System in Celiac Disease Development.
So first of all, I really appreciate inviting me and then kind of providing really kind of great funding for the study. The title of is celiac disease the microbiome, but my study is not actually doing the microbiome. Actually, much more doing about using serum samples and then detecting about what kind of host responses against microbiota. Because of that, I just kind of introduced some kind of summary of microbiome in celiac disease first, and then I'm gonna show some our data and what is we are looking for, what we found from that. Okay. So disclosure is I really appreciate the Beyond Celiac Disease Foundation about supporting this one. This gut microbiota, I see about we know about diverse community and of microorganisms in residing gastrointestinal tract. And then this is unique. Each individual has unique systems because of some genetics and because of environmental factors, including diet. And generally, maybe by the age of 3, the gut microbiota may be stabilized, and they will still maybe the sensitive because of their lifestyle and then host factors. And host factors that include immune system and nutrient and metabolism of our protection or maintenance of the gut mucosal barrier. And health intestinal flora consists of these kind of 6 the big phylum categories. I think recently they changed about the actual phylum, the name, like a Firmicutes, they changed it to Bacillodata, and some kind of things. Maybe you can look at that. The phylum, the name is all changed recently. And genetic of celiac disease and microbiota is quite important because there are many studies using celiac genes like DQ2 and DQ8, and they showed about differential early gut microbiota composition in infants with celiac gene and compared to individual low gene risk. Some days, I'm gonna show this next slide about show that. An infant with celiac gene HLA-DQ2 actually showed compared to low risk infant, which is non-HLA-DQ2 or DQ8 gene, they showed high proportion of Firmicutes and Proteobacteria. I'm still gonna using about old term, not a new term because it's not familiar with me. And then low proportion of Actinobacteria can be shown and in high risk celiac gene infant. And also high risk celiac gene infant has showed decreased bacterial diversity that are shown in the kind of literature. And this is the kind of paper based on the published in Gut 2015, and they comparing about DQ8, DQ2 and 8 infants and compared to DQ2, they're going to show that increased Firmicutes and Proteobacteria and decreased Actinobacteria. They're going to shown in there. And also the same group looking at about 4 months and 6 months. And then when you look at that, the diversity and then celiac gene showed kind of an increase over time, and then showing about a little difference about this control. Control has kind of be the same and similar, but they showed about increased the diversity in over time. And this is just over a summary, over a difference in gut microbiota and celiac disease. Many studies and most studies showed low abundance of beneficial bacteria that include Bifidobacterium and Lactobacillus. And also they are showed about increased abundance of member of Proteobacteria, which could be maybe including Pseudomonas, either duodenal biopsy or even stool samples of children or adults with active or limited celiac disease compared to controls. And Proteobacteria like Pseudomonas data, which is kind of new term, were linked to the persistent symptom and despite celiac patient following a gluten-free diet. However, actual recent review paper reviewed all systemic analysis that showed very contradictory result. Because of many studies, actually small sample size, and then because really, really hard to understanding what the composition of microbiota, there are many methodology difficulties. I think it's next speaker we're gonna presenting about is really difficulty about measuring the methodology of microbiome analysis. And then how about the immune system? Immune system is really good. And then because gut flora and a critical role in function of both innate and adaptive immune system, this is where everybody know and already kind of know that. But how can you just kind of in interaction between our host immune system against gut microbiota? That's kind of more challenging question. So gut microbiota could upregulate pro-inflammatory or downregulate anti-inflammatory mediator metabolites. And some study shows about beneficial bacteria like bifidobacterium and showed anti-inflammatory properties, and then maybe through IL-10 and lead to inhibit production of TNF-alpha or interferon gamma. The regulating balance between CD4 and CD8 T helper cells. And also, additionally, some studies and showed abundance of bad bacteria in like Bacteroides and E. coli and Pseudomonas. Could it be that related to increased intestinal permeability, or maybe increased inflammation of the mucosal membrane? And then there should be have a host factors, because host factor is not going to be much studied on microbiota. And there are one maybe secretory IgA, which is significantly low level in infant who develops celiac disease. Kind of publish the data. Maybe they have an antimicrobial peptide, which will increase AMPs in pediatric celiac patients. And the mucosal layer and mucin, they have kind of related significant increase in small bowel mucosal celiac patient. And how about IgG? IgG is also kind of another good impact about interacting with the microbiota and controlling microbiota. Some studies and showed IgG can coat most intestinal bacteria, showed in patients with IgA that are based on IgA deficiency studies. So this study is based on IgG that kind of using about is children with atrophy or asthma. This means children has allergic children. When you look at the data about this is 0 months, which is at birth, and 6 months and 12 months, 12 months and 2 years and 7 years, and they showed about comparing about control and allergic patient. There are some show the difference of immune reactivity, which is mean IgG activities is increased in some formicutes or some other genes. They got this data, I think, with UAB group. We just collaborate with them. We're using this microbiota antigen chips, and we look at our data. And then this is little busy slide. Maybe I need to kind of explain a little bit about host antimicrobial IgG. Could be depleted in the target in the gut microbiota. Okay. So the first A, a kind of figure A showed we can tell looking at about IgG kind of coating the fecal bacteria using about pieces and then put it IgA and whether they can be reacted or not. And they showed a non-IBD group has low. And then compared to ulcerative colitis or Crohn's disease, showed about really kind of highly IgA caused in that bacteria and the feces. And then based on using that one, we're using a health donor, surrogate fecal community, using about surrogate fecal community, and then samples from patients, and then mixing together, and whether some bacteria is coated with IgG because of their condition, disease condition, and then we can develop some IgG scores based on this description. And this is just kind of very brief kind of introductory slide. When we're just comparing about fecal community and with IgG score, this kind of showing this blue color and the red color and indicating about many bacteria deficiency is very correlated with IgG immune reactivity. When you look at the kind of right graph, which is G, looking at about 10, 20 taxa, which is most depleted, and then they're gonna show about IgG score will really increase in those groups. It could be indicating about some depletion of bacteria, could be good bacteria or beneficiary bacteria. It may be depleted because of this systemic immune reactivities. And this is kind of our the concept slide. And because many studies actually using about pediatric samples, and then we are using about adult samples, whether because the celiac patient can occur even at adult ages. Why? Why happens? But this is maybe they have ongoing gluten exposure all the time their life. They may have some kind of triggering effect or some kind of augmenting effect. It could be happen in that time of period, and then some suddenly they can maybe develop some destruction or even their symptom is occurred. And how about can you look at about autoantibody changes? And at the time of early phase or preclinical stage of disease, well, we're gonna looking about antibody against gut microbiota using this concept. Okay, and then this is kind of our aim about utilizing DOD, which depends on the preclinical sample, and we can showing about changes of TTG IgA titers and immune recognition of gut microbiota based on 25 small gut microbiota. Fortunately, and this kind of DOD cohort has such a kind of great system. They can medical record is available, and also the serum sample they collected because of every 2 years of HIV testing. So every 2 years they do HIV testing. After remnant of serum sample, they're stored in the freezer, and then we can capture this medical record and plus serum samples together. And this is kind of sampling time point. We collected the samples just before diagnosis, which means they have some ICD-9 or 10 code of celiac disease. And then we just kind of retrospectively collecting serum sample about 2 years and about earliest available sample, which is meaning some samples available at the time of military entry, which is we consider is really, really healthy at the time of military entry. They don't have any symptom because they start to kind of work in military as a personnel. So we have about 159 celiac patients based on this ICD-9 and 10 code, and based on 2 medical encounters, and then we just using this serum sample and we measure this antimicrobiota antigens. This is all kind of sort of what 25 microbiota antigen we used. And I just kind of briefly categorize about which is bacteroidetes and the Firmicutes flagellin, because Firmicutes flagellin is really important to and having adapting immune regulation because of the aseptic route and the Firmicutes and other and proteobacteria. And interestingly, about 159 celiac patients, it's about 10 years ago, they showed still have already positive the celiac disease. This is shown in here. Okay, then next. Showed in here about this kind of blue column is representing earliest available serum cell, which is even 10 years before the diagnosis of celiac disease. They already had positive. The titer based on Innovatase, which is 4 years reference level, they already titrated 60. And at the time of 10 years ago, maybe very similar to, close to military entry point. The some group like showed up a persistent positive group. So another group showed up a seroconvert group, which is meaning in the negative at the before, and then they got turned to positive later. So we're using this data, this serum sample, and we tested this 25 microbial antigens. And this is we found, and we're comparing about controls, and then they showed bacteroidetes, and then we comparing these 3 groups, persistent and seroconvertant control group, and they got some difference about Firmicutes group, which is going to little increase and then control, but a little decrease in their celiac patients. And this is kind of Prebacter group also increase in control, but when decrease in about 10 years before early diagnosis. But interestingly, the permaculture flagellin, when you're comparing the controls, and over even 10 years ago, they already showed strong immune reactivity against the flagellin antigens. And then we look at about this just before diagnosis, which is just before, which is TTG positive one. And then I think others, most important part is maybe permaculture flagellin group still have showed a strong immune reactivity and against permaculture flagellin group. But others, and they are increasing and decreasing based on their time over time. And right now, this is just kind of preliminary data. We're going to be looking at more detailed data and later. And also, we have another data set because that's kind of looking about progression of immune reactivity, which is again is gliadin and tissue transglutaminase and gliadin tissue glutaminase complex based on peptide microarray. And then using about micropeptide chip and put it about all and like 12 moles from gliadin and TTG. And then we want to looking about interaction between, integration between these immune recognition and the food antigen, also these microbiota antigens together. And then maybe we can see some very interesting data later. Okay, in summary, in seroreactivity and the microbiota is at the preclinical stage of disease. In celiac disease, it's mentioned that seroreactivity again is specific permaculture flagellin and was significantly higher many years before the clinical diagnosis. Maybe strong reactivity, certain permaculture flagellin is remain higher, and then maybe we need to look at about this furthermore. In conclusion, and gut microbiome, bioabundant celiac is very maybe influenced by the factor of genetic environmental triggers. And then gut microbiota host is complex, and host immunity also impacting gut microbiota ecologies. And seroreactivity to gut microbiota can occur many years before the disease before diagnosis. And immune response to a certain gut microbiota could be important for the pathogenesis of celiac disease. And this is another slide, and thank you so much for inviting me.
Thank you. Okay, thank you very much. Our last speaker for the morning will be Sónia Pereira at the Institute de Política de Lira in Portugal. And the talk is entitled The Challenges of Microbiome Analysis and Fibre-Foraging Species. Thank you.
Thank you. Thank you all for being here. Thank you, Beyond Celiac, for inviting me and, more importantly, for funding my research project, which is named Celiactive Project, Translocation, Inflammation, and Virulence, Dissecting Mechanisms of Gluten Microbiota Interactions in Celiac Disease. So, of course, we have to always present our conflict of interest. So the purpose that, and if we have some bias on our research, and so these are my conflict of interest, my 3 kids. So the thing is, for over the last 16 years, I've been living with celiac disease. I'm not a celiac patient, but my 2 kids, the youngest ones, that you see there in the photo, they have celiac disease, and the boy doesn't. So the 3 of them have the genes, and only the girls have the disease. So, and even between them, one has more severe symptoms than the other. So I wondered why. And so as a microbiologist, I started thinking maybe, so this is the terrible 3. The other terrible 3 are at home, and now they're grown-ups, which make them even more terrible. But regarding the disease, this is the terrible 3. So we know the trigger, it's gluten. We know the genetic predisposition, it's these 2 genes that we all know about. We know the autoantibodies that makes this disease an autoimmune disease. However, only around 3% of all the carriers develop the disease. And in general, we also have, in the literature, we know that around 40% of people around the world have the genes, but only the carriers, only 3% develop the disease. So we also know that some patients remain symptomatic even if they are under a gluten-free diet. And on the other hand, we may have people with the genetic predisposition, with elevated titers of the autoantibody that remain asymptomatic. So all these questions lead me to a possible unknown for participants. And again, as a microbiologist, I think that microbiota may have an influence on and may have something to say on this disease. So just a brief note on that. So we are a person, of course, but when you look at the number of our cells, each and every one of us in this room and in general, we have 10 more microbial cells than cells of our own. And if you look at the genes, more than 100 more microbial genes than human genes. So microbiota may actually influence a lot or not the disease. So we also know that we depend on microbiota in many ways. It's important to mature our immune system, of course, but it also helps prevent infection, and it's involved in vitamin production, etc. So metabolite elimination, and in digestion, of course, around 90% of our feces, the weight of our feces is bacteria. So, and it can also regulate our humor, our mood. So microbiota influences a lot. So what about celiac disease? Huge information on that also, and several studies report gut dysbiosis in celiac disease patients. And we also know that inflammation and bacterial translocation can increase the disease response, but what are the mechanisms involved in this? And how can knowing these mechanisms can help us find different solutions for celiac disease patients. So in my Celiactive project, the first act was to, and as a microbiologist again, was to understand the state of the art in celiac disease microbiome studies. So I did this review, which my colleague already mentioned in his presentation, and briefly, in starting, so I focused on studies in celiac disease patients and gut microbiome using next generation sequencing, which is the technique most frequently used in the third millennium. So from January 2000 to July 23, which was when we did this review. With no restriction on age, gender, etc. We tried to gather the most number of papers relating to this. And two-step approaches or culture-based studies were excluded. So at the end, we started with, I'm short-sighted, 489 papers, but then all the procedure led to a total of 48 studies being analyzed. Of course, I'm not going to dwell much on the work of other colleagues, of course, but just the brief introduction. So the majority of studies are currently being made in Italy and North America. Of course, after Italy, we have Finland, Spain, and the United States as the countries with more research going on gut microbiome and celiac disease. Some studies were prospective, others were cross-sectional. Some studies focus on children, others on adults, only 4 on both, and 4 didn't present the population they studied. So the majority was on feces. Some studied duodenum. So there's a huge variability of approaches in this area. So even the number of participants ranged from 6 to more than 1,000. The majority tested both sex. Ethnicity was only mentioned in 2 studies. So one thing that struck me was the inclusion and exclusion criteria, because 23 of these studies apparently didn't mention them. So that's that. And just to give you the conclusions, so I start with the discussion section. There was no universal correlation between a specific species or genus or phylum and the celiac disease, regardless, even looking at the patients, regardless age, the gluten-free diet status, etc. And another thing that also was striking to realize is that the information on those papers was not completely enough to allow to reproduce the work done. So this is related to the huge technicality associated to the studies and everything. So since the patient recruitment until the data analysis, there's a huge variability, but it wasn't expressed that specifically in these papers. So very briefly, this is, of course, you can look at the paper. So this is results regarding the film analysis inside of each. Only a brief. You have other tables in the paper, but as you can see, and what I would like to highlight here is that. So for example, so looking at the most prevalent phyli, so the big groups, you can see that in newborns the phyli is more, it's higher than the controls, and in children some studies say that they are equal between celiac disease and controls, and in adults some are higher, some are equal, some are decreased, which makes sense because as life goes on, life gets complicated or more complex, not complicated. So the same thing happens also with our microbiome, with our gut microbiome. It also gets more complex as we grow up. And this is an example for other groups of other phylum, in this case the bacteria. That is the same thing happens. So in some studies it was decreased, in other studies it was increased, comparing celiac with the controls. And also again the same thing. Some decreasing, others increasing. So that's that. So this was the first act. So the Act 1 in the project. The Act 2 was trying to address a novel perspective compared to what was already done regarding celiac disease and gut microbiome analysis. And so this is our original paper, recently published. We voluntarily invited celiac disease patients and their first-degree relatives. And why did we do that? Because the first-degree relative, and we had that care on our selection, so at least we try to reduce some variables. So being first-degree relatives, they are the most genetically related persons, looking at the human genes, and also in terms of diet and the house they live and the things they do, and well, they grow up together. So that's why we aimed to this celiac disease patient and the first-degree relative. So the inclusion criteria and exclusion are over there. And moving on, what did we find? So this is also the socio-demographic characterization of the patients and their first-degree relatives. We went to, so Portugal is a tiny country, but we went across country to collect these, to recruit volunteers and collect the fecal samples. So when we were, so even still in the demographic of the volunteers we were able to recruit, there was a statistical significant difference in the nutrition supplement intake. So we're not looking at gut microbiome yet, but only the features that we collected. And we collected also information about their dietary habits. And so celiac patients, there was a statistical difference between celiac patients and their relatives regarding nutrient supplements. So celiac patients take more nutrient supplements than non-celiac relatives. In terms of microbiota results, so alpha diversity regards the diversity, so the composition of each sample, each microbiome sample, so or the abundance of different species or whatever phylogenetic level we are looking at. So there was no big difference between the celiac patients and the relatives, but the relatives had not with statistical evidence, but a higher diversity. That's what you see there in the graph with the arrow aiming. So a large number of species on the first-degree relatives. Regarding beta diversity, which compares the group of bacteria that a person has with other factors. So it's like an ecosystem. That's the rationale behind. So the gut microbiota was influenced by sex, age, and the body mass index of the volunteers. And so we included that information in the models that we afterwards we generated. So we also wanted, because this celiac disease and the first degree relatives, we wanted to confirm that our approach made sense. So this kinship approach, testing relatives, and we realized that in fact, so the general microbiota of family related was more similar than the celiac versus putting all celiac patients in one group and comparing to the first degree, to the non-celiac, not the first degree relative. So to the non-celiac, it was more diverse. So gut microbiota of relatives are similar than regarding the disease by itself. So I think we kind of proved that this kinship approach was okay and that our results were valid. This rationale was valid. So what did we find? Oh, sorry. So what did we find? We find that first degree relatives, this is just the highlight of the species that we found increased in the first degree relatives. These are the ones increased in celiac disease. And so we understand that celiac disease patients had lower rates of fiber degrading bacteria. And so we looked at, when we realized that, we compared with the dietary patterns, and we realized that some species have a negative, so some, not the species, the habits that people have apparently impact the presence of some species. So as a highlight, this Rhamnoclostridium serum was negatively affected by the nutritional supplements and the positive effect. So there was an increase on this species if a person ingested more vegetables or have 3 servings a day of meals. But celiac disease was on celiac and non-celiac, but they were lower levels of this species. Akkermansia mucinifila, the same thing. And just to give an highlight, of course, we can never forget that although they are relatives, they have different diets because the non-celiac wasn't compliant with a gluten-free diet, okay? So we also have to keep that in mind when comparing results. So finally, and in the discussion briefly, these species that were different, so that were reduced in celiac disease patients, the literature tells us that are already associated with glucose metabolism control, for instance, or improves gut barrier, and they are decreased in celiac disease compared to the first degree relatives. Also Akkermansia, the same thing happens, so it has a positive contribution in inflammation reduction, which is highly important in several diseases, and its depletion, of course, is involved already in type 2 diabetes, etc., so not to mention them all. So the final conclusion is, in fact, celiac disease have a decline in fiber intake, looking at their dietary patterns, and also the correspondent fiber fermenters. And of course, until we have a cure, maybe it's time to reinforce the need to have better or more or fiber-added gluten-free products, so that celiac disease that are prevented from eating all type of foods can counterbalance this lack of fibre-foraging. So that's that at the moment. Thank you very much. I would like to appreciate my research team, of course, all my collaborators and those that will collaborate in the next steps of the projects, and thank you all once again.
Sorry. No. Thank you very much. So we have a Q&A and then lunch. So it's okay. I will ask one question. This is mostly for Dr. Choung. In terms of, screening is a real big issue. And it's mostly focused on pediatric screening. But from your work, what would you suggest? Because you're now looking at adults.
Yep.
And
So I think it's 2017. The preventive service kind of announced about there are not evidence, no evidence is not, or limited evidence, and lacking of evidence to kind of screening for the general population. That is, already announced 2017, and at that time we just kind of make a commentary about that. And interestingly, recent our study showed someone has positive TTG-IgA, and we're looking about following about what they're gonna just kind of staying on the gluten consuming based on our kind of community database. Among about 20 to 30% population of CDA, celiac disease autoimmunity, which means seropositive patient, actually turned to negative later, about 5 years later. Those kind of people we may need to find out. It could be the potential celiac patient, and it could be maybe reverse their autoimmunity, why that happens. So maybe that's could be answer about whether we're gonna do screening. If we're gonna do general screening, then we might maybe unnecessary people may on a gluten-free diet, they may be obtain a good kind of chance to reversing their autoimmunity. So right now, I don't have any answer for that, whether it's really kind of recommendable general screening or not, but still have there are opportunity to study about those populations.
Thank you.
Thank you both for the interesting talks. I want to ask you, Sonia, if I understand correctly, you mapped the strains and then connected them to the functions. But did you consider doing metatranscriptomics and sort of analyze just the pooled bacterial genes to see if there is genomic functions that are correlated with celiac versus controls?
Yeah, so we didn't do that exactly. We did the next generation sequencing of a specific part of the bacterial DNA, and then we compared it with the information we collected from the patients. The metatranscriptomics would be a plus, but we weren't able due to, not regarding Beyond Celiac, of course, but due to funding constraints, because it's too much more expensive. So we have to start in somewhere. So we decided to start with this, only comparing the gut microbiome with information collected through a questionnaire to the patients. Metatranscriptomics will be the next step, hopefully.
Thanks.
Yeah.
Kati?
Hello, Kati Juutiustalo from Tampere University, Finland. Thank you for your both, but I have a question for Roxanne Choung. You have fantastic pool of data from the DoD. I have that kind of stupid question. How intense the inflammation needs to be so that you can detect that with your from the HIV tests from the blood? So because if you have only a small or minor infection, will that be detected with your methods?
So your question is, how can we detect?
What is the level? So that how sensitive is your test that when you see that there has been an infection?
So I don't know about whether HIV testing is, because I have no idea about what actually they are sensitive to HIV testing. But right now our study is using those serum samples, remaining serum samples after testing, and then we just kind of looking at about whether there are any signature for related to the celiac disease or not. This one of them is just kind of looking at the microbiota antigens. I think this we can collaborate with the Chuck Ellison's group, which is kind of they published lots of data in the Crohn's disease, showed the really increasing activities against flagellin and flagellates. That's, maybe it could be one of the pathogenesis of Crohn's disease too. And also the interesting is celiac disease, and the similar features we can see the flagellin and against antibody against flagellin also increase in celiac disease too. Whether they're going to be causal effect or not, but that's going to have to be studied more.
Okay, so one last question, and then we'll be ready for lunch.
A very quick question to you both. What are the resident bacteria in the duodenum? Because the resident microbiota in the duodenum, obviously the numbers of organisms goes up enormously as you get to the colon, but that area straight after the stomach, there's relatively few organisms. I presume it's known by now What's resident there?
Yeah, so I'll translate that to the microbial signature
Yeah.
of the disease. Yes.
No,
so-
well, from biopsies. ideally,
Yes.
yeah.
So the review that we did didn't lead us to a specific microbial signature. That's the thing. And even, for instance, fecal transplantation, which I believe it can be a possibility for this disease or others, of course, because I think it has immense potential. The question you made is very interesting because it's truly what I believe. So we have this residual microbiome inside. Even if we clean, and for instance, to do a colonoscopy, we have to clean the entire intestine. And if we just leave it there to rest, that residual microbiota will come back again. And in fecal transplantation, the same thing happens. So you do the transplantation, and you can shift a little the gut microbiota, but then the residual, the signature, will arise again. So that's that. But I don't have that a correct answer. The only thing I can say is that we have some information, some guidance from all this work that is being done, but no microbial signature is yet. We didn't get to that conclusion yet.
So my answer is because I'm not a microbiologist. But I think it's in our lab is looking about microbiology too. But it's oral microbiome, which is really kind of common and reside in the small intestine, especially in the Preclinical group. Preclinical group is really kind of increased. And then recently, in our lab, we actually found isolated from celiac patient, PSD cola, which is called Preclinical cola, which is that showed really kind of promising beneficial impact for even delay or suppressing immunity and in autoimmune disease. That is kind of published in MS and rheumatoid arthritis, some other autoimmune diseases too. So Preclinical group is much more common, which is kind of very similar to oral cavity and microbiome.
Thank you very much. So this is the end of the morning session.